the system automatically applies some brakes. So that will be the last step. So what tools I'll be using in this research will be simulation software will be using. So mainly I will be going with the PTV WISM software because that's based on the 3D simulation that's based on it shows all all the scenario 3D. So this is the most advanced software which I'll be using. Some geographical information systems, artificial intelligence and machine learning that I already informed you regarding transect and queuing theory approach will be also used. And for optimization of the shortest path, we will be also considering the Recastras algorithm. So some virtualization tools will be also used. And again, some surveys and questionnaires will be also used to collect the data about the traveler information, traveler behavior, preference, attributes, etc. So at last, the expected outcomes of this research would be accurate understanding of effectiveness of ITMS that we use to develop reducing congestion and improving road safety in urban areas. And it will also, provide insights about the challenges and limitations of implementation of ITMS and their best practices. And it will also recommend the optimization of ITMS to ensure the maximum effectiveness in reducing congestion, and it will definitely improve, road safety. So this is all about my research topic, comprehensive analysis of ITMS. If anyone having any questions.
Directly, Ashmat, am I audible to you?
Yes.
Ashmat, what is the field of your sir?
Can you just repeat your question, sir?
What is your research field?
Research field, actually my thesis is all about this ITMS, implementing a comprehensive approach of ITMS to reduce congestion.
You are from computer sciences?
Yeah.
How will you correlate your work with the theme of this conference?
Sir, the main idea of this, the theme of the conference is development of the, sustainable development. I guess.
How will you correlate your work with the theme?
Sir, I guess if we can minimize the traffic congestion, we can develop such systems. That will also lead to a sustainable development of the society. If the accidents will be if we develop such systems that will pre-detect the accidents, and that will also optimize traffic flow, and that will also reduce the congestion, and that will also maximize the flow of the. Likewise, we face various issues while we we can't make a way for the emergency vehicles. Likewise, they get stuck in traffic flow. So if we consider that scenario, we can match that scenario, and that will be also beneficial for them as well. That will also lead to the benefit of the sustainable development. If we make a way, if we reduce the congestion, and if we make a free flow for the emergency vehicles, we can just save patients. If we can just, if so, I hope you got my point, right?
Okay. This is your original research or just review of the earlier work?
So it's just an analysis. It's just an analysis paper. So we'll be undergoing in this research in the next phase.
And what are your own findings?
So you can just repeat. Your voice is breaking.
Okay. Sufficient.
Okay, is it any questions? So we have finished your poster presentation. Now the next one is Naveen Badial from NIT Jalandhar. Are you there?
Naveen
Badial for the poster presentation from NIT Jalandhar. Then the next one is a Shishpal Reddy from Rohtak. Shishpal Reddy from Rohtak for the poster presentation. Are you there?
Shishpa, are you there? Next one.
The next one is Munisha Mahajan from Career Point University. Munisha Mahajan. The next one is Renu Bala. Are you there? Renu Bala?
Yes, good morning, ma'am.
Good morning. Okay. You will present your screen and share your screen here.
Yes, ma'am.
And you have only 5 minutes for completion of the poster presentation.
Yes, ma'am. Yes. Good morning, ma'am. Can I start?
Yeah.
Yes. Today's my topic is importance of pollinator and pollinizer in fruit set, quality and yield of fruit crops. And the introduction about the pollination is transfer of pollen from an anther of a plant to a stigma of a plant of flower. And pollen grains are immobile. They cannot reach the stigma by themselves, and external agent is required for this. It can be wind, water, animal, and gravity or growth contact. And there is a two types of pollination: self pollination and cross pollination.
Hello, hello. Sorry to
Self pollination.
interrupt. Renu, sorry to interrupt. Can you show the big screen?
Yes, ma'am.
Please, you can.
Yes. There are two types of pollination: self pollination and cross pollination. Self pollination, the pollination of a flower by pollen from the same flower or from another flower on the same
तो आप
plant.
इसको लैंडस्केप में कर सकते हो?
What's a?
ये मोबाइल में चल रहा है ना, तो लैंडस्केप
Yes,
में आएगा?
I can try. Is it okay, sir?
हाँ जी, कुछ ठीक है.
हाँ जी. A pollinator, next is cross pollination, is the process of applying pollens from one flower to the pistil of another flower. Then next is pollinator. Pollinator is anything that help pollens from male part of the flower to the female part of the same or another flower. A pollinizer is a plant that provide pollen from often essential for fruit set or development of fruit. And there is importance of pollinizer or some pollinators picture. And pollinizer, importance of pollinizer can improve the pollination fertilizer mechanism, which leads to increased production owing to larger and better shaped and quality of fruit. It can enhance the number of fruit per tree, ultimately increase the yield. There are some effects of apis cerana on yield of mango. There, in which the highest value in bee pollination, three at the rate 7 hives, 84.75 is the highest value in yield of mango due to the effect of apis cerana. And effect of different time of pollination on yield component of date palm. Uh, it it uh, on different time, just like 6 to 8 or 8 to 6, and on a fruit set production or yield of date palm. And there are some pollinizer, amla, ber, pomegranate, bale, fig, falsa, and custard apple. And my conclusion is that pollination is an important ecosystem services and honeybee is valuable pollinator. It leads to fertilizer, successful seed development and fruit production in fruit crops such as lychee, mango, date palm, apple, etc. Apis mellifera on the apple increase in number of fruit panicles and its weight. Thank you so much. Am I audible?
Yes. अच्छा रेनू, what is the significance of your search?
Yes, sir.
डॉक्टर रईस आप करिए बात.
सर क्या बोल सर फिर से रिपीट करना.
डॉक्टर रईस.
Okay, Renu.
Yes, sir.
Yes, you have presented this information in a very simple way. Can you just tell me different types of cross pollination?
Cross pollination, entomophily, and through wind, insect, and hydrophily through air. Sorry, water.
What is getonogamy?
So what?
Getonogamy.
Sir, it is the type of self pollination, and transfer of pollens from stigma to anther, same flower,
Stigma to anther.
but genetically different.
Stigma to anther.
So what? Anther
How
to
do you
stigma to
define it?
anther. Yes,
How is
sir,
it
in
different from
genetic.
autogamy?
So what?
Autogamy differentiated from autogamy.
Autogamy is a type of self pollination, and in this pollens transfer from anther to stigma on a same flower of same plant.
And getonogamy.
Heteronogamy, sir, इसमें genetically different होता है plant. Is just like genetically just like cross pollination.
Just like cross pollination. But it
Yes,
is a
sir.
type of self pollination.
Self
You are saying?
pollination. Yes, so self pollination is the two types. Uh, Autogamy or heteronogamy. Yes.
Then what are your ultimate findings in your research?
Sir,
Can you
there is
conclude in few lines?
सर इसमें यील्ड में इफेक्ट पड़ता है जब हम क्रॉस पोलिनेशन करते हैं. मतलब क्रॉस पोलिनेशन जब होती है जैसे कि विंड के थ्रू हो गई या फिर एंथर जो है वो खुद नहीं, वो इमोबाइल होते हैं तो वो खुद नहीं स्टिग्मा तक पहुंच पाते हैं, तो ये एक्सटर्नल एजेंट्स जो हैं वो कुछ
ये
हम
तो
यूज करते हैं तो
ऑलरेडी प्रूवन है ना. All this is already proven. What are your findings?
यस सर. सर मैंने सर्च पेपर पढ़े हैं सर, जिसमें मैंने खुद रिसर्च नहीं करी है इसमें कि उसमें सर जैसे कि
मतलब रिसर्च ओरिएंटेड ना.
यस सर.
आप कुछ प्रेजेंट कर रहे हैं, रिसर्च प्रेजेंट कर रहे हैं,
यस सर.
सिंपल आ रहे हैं, तो दिस इज फॉर लोअर स्टैंडर्ड. आई थिंक
यस
you have to present something new. Your proposal.
यस सर.
What you are going to find out.
यस सर.
It is here.
यस.
That is no called research.
यस सर.
Okay.
ओके सर.
Going in better way.
यस सर. थैंक यू सर.
Okay, the next one is on Mittu Bharti.
Mittu?
Mittu Bharti.
Mittu Bharti, are you
Yeah,
there?
good morning, all of you.
Good morning. You have to share your screen.
Yeah, ma'am. Okay, I am audible. Am I audible?
Yes.
Yeah. Okay, good morning, all of you. The topic of my poster presentation is waste heat recovery through flexible films of conducting polymers. Now, my research is focused on thermoelectricity. Thermoelectricity is the power generation which converts temperature difference or uses ambient heat to convert into useful power using Seebeck effect. Seebeck effect was given by Thomas Seebeck whenever a temperature gradient is established across a conductor or semiconductor. Charge carriers from hot end move towards the cold end, and as a result, a voltage is generated which is known as Seebeck voltage, and this voltage can be used to power small scale miniature devices. These days, thermoelectricity is very much focused as a clean and green energy option and to harness natural as well as waste heat. Moreover, the thermoelectric power generation can be useful at indoors or in remote locations where other energy conversion technologies such as solar energy are not feasible. Now, conducting polymers, they offer many advantages. They are non-toxic, they are flexible. Moreover, they can be solution processable, and therefore they can be deposited in form of films, flexible films, and these flexible films can be wrapped around irregular objects such as human body also, so to harness waste heat or to recover waste heat. Conducting polymers, they are having low thermal conductivity, they possess amenability towards doping, and they are nature friendly. Therefore, they can be considered as promising thermoelectric materials to recover waste heat or harness natural heat around 100 degree or low temperature, less than 150 degrees Celsius. The present work therefore intends to investigate the role of conducting polymers as promising thermoelectric material for waste heat harvesting. Now, in our lab, since polypyrrole films were grown by photopolymerization route using UV light, and P-dot PSS film were directly deposited, drop casted on Kapton substrate. These films were characterized through various techniques like XRD, SEM, AFM, TEM, Raman, and FTIR, and thermoelectric properties like electrical conductivity and Seebeck coefficient, Seebeck voltage, were measured through the customized lab-made apparatus. In results and discussion, what we find that thermoelectric properties of these polymers can be tuned by adding various dopant, by varying their concentration, and by addition of inorganic component. Later, we also developed some prototype thermoelectric power generator based on these conducting polymer films. In case of polypyrrole silver composite films, they were grown by photopolymerization route. We varied the content of silver, keeping the concentration of polypyrrole fixed, and we find very interesting phenomenon that in spite of adding the silver particles which were having high thermal conductivity, the thermal conductivity of these composite curtailed down by the 2 order. This reduction in thermal conductivity is attributed to scattering of phonons by the interface, which organic-inorganic interface. As a result, organic-inorganic interface can be created intentionally to reduce or to manipulate the thermoelectric properties to have high thermoelectric efficiency. As a result, highest ZT thermoelectric figure of merit 7.4 into 10 minus 3 at 60 degrees Celsius was obtained in these films. We also used 6 ppy AG strips, and by
electrically connected in series and thermally in parallel, and this generator provided output voltage of 6 millivolt or power of 30 picowatt when the temperature gradient of 65 degrees Celsius was obtained. Now, in case of P dot PSS composite film, we mix the bismuth antimony telluride, which was synthesized in our lab, with the P dot PSS solution and simply drop cast it. From the figure, you can easily see that the first figure is substrate adherent on Kapton, and the second figure is just peeled off due to the addition of bismuth antimony telluride. Such freestanding, or we can say self-supporting film, can be used to fabricate wearable generators, which can be used to trap body heat or the heat of irregular surfaces. These days, many research are being conducted to reduce such type of materials in form of thread so that thermoelectric fabric can be made. Now, conductivity of P dot PSS on addition of bismuth antimony telluride got improved, and as a result, highest power factor was obtained. Now, in conclusion, I can say that thermoelectric power generator based on these conducting polymer films can be useful for waste heat recovery. Moreover, flexible films manifest potential of harvesting ambient heat of irregular hot surfaces. And by combining more and more such P and P or PN couples, like in commercial devices, the output voltage can be further boosted for practical application, such as powering the miniature electronic devices. Now that's all. Thank you. Hello.
Hello, Mulu.
Hello.
Dr.
Am I
Jesh,
audible?
Yeah.
Dr. Jesh,
Hello.
Am I audible?
Yes.
Okay, Mitu, is this your original research?
Yes, sir. I did my PhD on under faculty improvement program from BARC.
Okay. What are these thermoelectric polymers? Your research is on polymers, I think.
Yes, sir. Basically, my research on investigation of thermoelectric properties of conducting polymers. These are the original normal polymers.
You are not clearly audible. Sorry, you are not audible.
Excuse me. 1 second, sir. Sir, from my side, it is okay.
We can't hear you.
Hello? Sir? Hello? Hello?
Okay, that's good.
Hello?
hello, Dr. Jesh.
Actually, we can't hear the candidate properly here.
Sir, from my side, it is very much okay. I can hear you and my mic is working.
You are quite audible, ma'am.
Okay, then switch over to next candidate. It's not well, appreciable.
Okay, thank you. Now the next one is a
Sir, should I leave?
Yeah, definitely.
Okay, thank you. Thank you so much.
Kirti Sharma. Kirti Sharma, are you there?
Kirti Sharma,
Kirti Sharma. For the poster presentation, Kirti Sharma.
Kirti Sharma,
Kirti Sharma for poster presentation. Are you there?
रिदम का करा दो रिदम
I again repeat the name of the poster presentation. Naveen Bayal, Shripal Rathi. Are you there? Naveen Bayal, Shripal Rathi, Munisha Mahajan, and Kirti Sharma. Okay, you are not there, so we have to complete the poster presentation session. Now we have to start the online oral presentation session. The first I start with the rhythm.
हेलो रिदम आपका नाम बोल रही है
Hello, Rhythm, are you
हाँ, अभी बोल रही
there?
है।
Next one is the Baljinder Kaur. Baljinder Kaur.
मेल आईडी
Baljinder Kaur.
Baljinder Kaur, are you there?
अभी तो कनेक्टेड था आपका तो फोन से कर लो फोन से दे दो प्रेजेंटेशन हाँ फोन से है आपके फोन में प्रेजेंटेशन नाले पंगे बड़े हैं कनेक्ट नहीं
Okay, now I start today. I start the next technical session. Dr. Suresh Kumar from Government PG College, Una. Are you there, sir?
एक बार डाल दो उसमें डाल दो कि भाई प्रेजेंटेशन स्टार्ट हो गई है
Nandlal.
ऐसे नहीं
पहले मैं डाल दिया
Nandlal.
दोबारा डाल दो
And the next one is the Savita Devi.
देखना इनका टाइम
Suresh Kumar.
11:30
Avinash.
Yes, ma'am.
Deepika.
Yes, ma'am. Yes.
Avinash.
Yes, ma'am. Avinash.
Yeah, Avinash, you are there. You have to start your oral presentation, and the time duration is 5 minutes.
Okay.
Now you have to share your screen.
Ma'am, is this visible? Is my screen visible?
visible
Yes, visible.
very much visible
Thank you, sir. So, topic of my presentation is human wildlife interaction, a case study of asiatic black bear in Chamba Forest Division of Himachal Pradesh. As Chamba district is northmost and beautiful district of Himachal Pradesh, it is located in the lap of majestic, beautiful Himalayan mountain range in Western Himalayan. This has total forest cover of about 24,000.
I'm sorry to interrupt, Avinash sir. Full screen.
Okay, ma'am. Okay, ma'am. Is this okay?
It's okay.
Okay. So district Chamba forest circle have 5 forest divisions: Chamba, Bharmor, Dalhousie, Churaha, and Pangi. Chamba forest division is expanded in about 4,230 square kilometer. This forest division has 4 forest ranges, that is Chamba, Lower Chamba, Upper Masroon, and Tikal forest range. Chamba forest division, as I am working in this forest division, having altitude of about 1,200 meters to 4,000 meters above mean sea level. Forested area ranges from lower elevation to high altitude area. Important source of livelihood. There is forest resources are important source of livelihood in this area. This is an important ecological and cultural asset of Himachal Pradesh, having huge diversity, faunal as well as floral biodiversity. As I am working on bear, Bear belongs to family Ursidae. Totally in world, there are 8 species of bears, that is Asiatic, American, polar bear, Himalayan brown bear, sloth bear, spectacled bear, sun bear, giant panda. 6 species occurs in Asia, 3 in North America, and in India, as far as India is concerned, 4 bear species are reported. That is Asiatic black bear, as I am working on, sloth bear, Himalayan brown bear, and Malayan sun bear. This is about the area of study, that is Chamba Forest Division. This is the map showing the distribution of Asiatic different bears in India, including Asiatic black bear. As far as these are 4 species: Asiatic, sloth bear, brown bear, and sun bear. Asiatic black bear is listed in IUCN Red List as vulnerable species. This is about the classification: phylum Chordata, class Mammalia, order Carnivora. Now about human negative human wildlife interaction. Uh, Habitat destruction, pollution, illegal wildlife trade, conflict with predator, wildlife trafficking, climate change, improper wildlife management. These are some threats to the bear livelihood. Asiatic black bear, protected species in India, plays an important role in maintaining ecological balance in including Chamba region. Deforestation, habitat fragmentation, expansion of agriculture has significantly impacted the bear population, led to increased human-bear conflict. These conflict primarily lies from resource competition, crop raiding, and potential threat. Crop raiding is the significant problem in the local farming community, and in nowadays or in current scenario, these human bear conflict have been increased in this area. Study area is this for Chamba Forest Division, and data collected from June 22 to December 23 from Forest Department, 1995 to 2023, and study carried out in from June 22 to 23. Research is based upon interviews of seasonal officials, forest workers, local people, herb collectors, moshella collectors, migratory laborers, nomadic raisers, shepherd, gujars, etc. Literature review also helped me a lot to find out the reason behind increasing incident of human wildlife conflict, or including in human Asiatic black bear conflict in the area. Material and method: informal interviews, socio-economic human black bear conflict, compensation paid by HP Forest Department, camera trap, hair traps. These are now about the research, origin of research problem. This research problem has been originated by increasing incident of human Asiatic black bear conflict in this area. This was necessary to analyze the effect of anthropogenic activity to change in the habitat of Asiatic black bear. Objective: to assess extent of human Asiatic black bear, signal dietary intake pattern, habitat use pattern of Asiatic black bear, threat to black bear, and importantly, mitigation strategy to reduce this. This is about these are camera traps of black bear in the area. Now about the result and findings. Significant population of black bear is there. Habitat loss and increased human activities have led to the increasing human Asiatic black bear conflict. Loss of property, livestock damage to agriculture crops, livestock killing, human attack. Now this is about the human.
Your time is finished. So what's your conclude?
Okay, ma'am. This is about the data of 2000 to 11 to 15, 20 to 23, and this is the cumulative data. Now, finally, I will be concluding about during 1994 to 2023, 75 human black bear conflict have been reported in the area, 53 injured, 4 deaths, 130 livestock killed or injured. Compensation of 18,84,339 was given to affected people, and total number of wildlife conflict as in area. Asiatic black bear were responsible for 125 livestock killings, as well as leopard killing, 558 killed by the leopard. Now I'm going to conclude. 65% of conflicts have been caused by Asiatic black bear as compared to the leopard. Now,
Okay, what's your finding after this research.
Okay, ma'am. Just uh, okay. This is about my finding. Over 55% of attack occur during dawn and dusk period. Mostly जब सुबह या शाम का समय है उसी दौरान ही होते हैं. 45% cases occur during summer and autumn and period of high human activity in agriculture field. Or the most important thing that most attack on human being occur when they are either going for work or returning after work. The conflict is further accelerated by the illegal trade in bear parts, leading to poaching and hunting. Bear hunted for their fat, body part, especially for gallbladder. So I think I should conclude. So there are some mitigation strategies.
Okay.
Thank you. This is all about my presentation. Thank you.
You leave the session, please, Avinash Pal. The next one is Baljinder Kaur. Are you there? Again, I told you. Baljinder Kaur. Then Deepika. Then Ritu Dhawan. Divya. Suresh Kumar. Sukhvinder Kaur. Vivek Kumar. Parvinder Kaur. Jagmohan.
Yes, ma'am.
Okay, you have to start. Jagmohan.
Yes, ma'am.
Jagmohan, share
Jagmohan,
screen.
share your screen and you have only 5 minutes.
Good morning, everyone. My topic of presentation is Descartes' method of successive approximation for fraction order initial value problem. First of all, outline, abstract, introduction, methodology, results, conclusion, references. In present work, we derive Descartes' successive approximation technique for fractional differential system in which the derivative has been taken in reasonable sense. We investigated the existence and uniqueness results of the present method. 2 numerical examples are given to show the efficiency of the presented method. Introduction. Classical calculus limited to the differentiation and integration of integer order, while the fractional calculus involves differentiation and integration of arbitrary order, that is of every real number order, for example, half order derivative, pith order derivative. Fractional calculus owes its origin to the question of meaning of the symbol dn divided by dx raised to power n if n is not an integer. In 1695, L'Hopital was the first who raised the question to Leibniz about the meaning of the symbol dn divided by dx raised to power n if n is equal to 1 divided by 2. Leibniz responds, an apparent paradox from which one day useful conclusions will be drawn. In the present time, the significance of fractional calculus has grown due to its capacity to broaden the scope of differential equations from integer to real number orders. Fractional differential equations have gained the attention of scientific community due to its importance in modeling of complex real-world problems in fractional order differential equations. Fractional order differential equations has application in various fields due to their ability to model complex phenomena more accurately than the integer order counterparts. Some notable applications of fractional order differential equations include electrochemistry, viscoelasticity, control systems, signal processing, biomedical engineering, fluid dynamics, finance and economics, earth science, etc. Indeed, many fractional order differential equations lack exact analytic solutions, necessitating the use of numerical methods to obtain approximate solutions. Numerical methods play a crucial role in tackling the complexity introduced by non-integer order derivatives or integrals. Some widely employed numerical methods for solving fractional order differential equations are fractional Euler method, fractional Runge-Kutta method, fractional Adams-Weisforth method, Adamian decomposition method, fractional finite difference methods, etc. Methodology. Picard's iterative method for integer order differential equations gives a sequence Ynx of approximate solution to the initial value problem given by this equation. Motivated from above work, we have extended the Picard iterative method for the fractional order system given by this equation. Here in this work, we have used the following definitions. First is Riemann-Liouville fraction integral. Let fx be the piecewise continuous function on the open interval 0 infinity, integrable on any finite subinterval of 0 infinity, and alpha be a non-negative real number. Then for t greater than 0, Riemann-Liouville fraction integral of fx of order alpha can be defined by the formula which is given by this equation. Similarly, the fractional order derivative of order alpha integration. Results. Our main theorem is let fxy 1 minus alpha order derivative of fx and curly f divided by curly y with the continuous function on some closed rectangle R and whose sides are parallel to the axis. Let x0, y0 be an interior point of R. Then there exists a real number h greater than 0 such that in the interval modulus of x minus x0 less than or equal to h, initial value problem given by 1 has unique solution y is equal to yx. In this theorem, we have proved that the sequence of approximate solution obtained from the formula given by this equation converges towards the exact solution. Here we have given the two examples to support our result. In first graph, we have plotted 20 iterations of Picard's iterative method by taking the interval 0, 3. As the number of iterations are increasing, the curves are superimposing on each other. That is, the curves are approaching towards the solution curve. In second graph, we have shown that the number of iterations, as the number of iterations increases, the approximate solution of this system converges towards the exact solution. Here in the second graph, the curve with the red circle is the graph of exact solution. As the number of iteration increases, we have shown in this graph that the curves are approaching towards the exact solution. Last is conclusion. In our mathematical analysis, we have successfully demonstrated the convergence of sequence generated by the formula represented by this equation towards a particular function, and this function serves as an approximation to the solution of fractional order system described by equation 1. The examples we have given shown that after limited number of iterations, approximate solutions are converging towards the exact solution. These are few references. Thank you. Hello.
जगमोहन हो गई आपकी?
Yes sir, जी sir.
अच्छा, what is Jagamohan? What is the initial value problem?
Sir, जिसमें हम we have defined the condition for initial point.
Okay. अच्छा, जो आपका जो method है, is it valid for second order differential equations?
No sir, अभी हमने सिर्फ फर्स्ट ऑर्डर के लिए ही किया है. अभी इसका पता नहीं है सर.
जो आपने प्लॉटेशन किया, कौन सा सॉफ्टवेयर यूज किया उसके लिए?
सर मैटलैब से की है मैंने.
ओके, बस ठीक है जगमोहन. Okay,
Thank you, sir. Thank you.
Okay, the next one is a Janinder Bhatti. Janinder, Pitambar Dutt, Nitika, or Sheetal. Are you there for the oral presentation? Now the next one is a Suresh Kumar, Vishal Mahi, Deepti Gupta, or Priyanka Thakur. Are you there?
Sukhvinder Kaur.
Yes, sir. Yes, sir. I have joined.
You have to start, please.
Okay, ma'am. Please let me share my screen. Am I audible, ma'am?
Yes, audible.
Is my screen visible?
अच्छा ये मोबाइल पर है तो इसको आप वो कर लो, लैंडस्केप में कर सकते हो?
मैं ट्राई करती हूँ सर.
देखो तो जरा.
हाँ सर, क्या लैंडस्केप में आया है?
हाँ हाँ आ रहा है.
ठीक है, तो क्या मैं शेयर कर सकती हूँ अभी सर?
अब इसको थोड़ा सा आप बड़ा कर सकते हो? हाँ ठीक है. अब
ठीक है सर?
हाँ जी अब ठीक है.
ठीक है, thank you sir. Good morning everyone. I am Sukhvinder Kaur and I am pursuing my past PhD from Sant Bhag Singh University from the Department of Botany. And today I have got the privilege to share my work on the seasonal parameters of physiochemical properties of an urban river of J&K, that is Balol. So I will like to share with you that for the present study, Balol Tavi, which is in the UT of Jammu and Kashmir, was selected to study its physiochemical parameters and the seasonal variation of the different parameters in the river body. In this regard, a detailed analysis of the physical and chemical parameters of water sample from the selected site was carried out from January 2001 to December 2001. Now, when we are studying the different water bodies, the water parameters, the physiochemical and the other parameters of the urban rivers, they are very, very screwed nowadays. Now this Banol Nala, which is an important tributary of the river Tavi, runs from the heart of Jammu city in J&K UT. It emerges from a natural spring near village Parmandal and flows through different villages and different areas of the Jammu before it finally joins river Tavi near village Nandwar, and from there it flows to Pakistan. So during its course, this rivulet, it receives various pollutants from Badi Brahmana industrial state and untreated wastewater and sewage from different residential areas, because this river really runs through the heart of the city, and it gets lot of sewage from the adjoining areas. So this river is situated this is the latitude and the longitudinal parameters of this river. For the present study, we selected a site near NITS Polytechnic on Jammu Araspura Road. It is a very busy junction, and here there are a lot of residential colonies. So this site was selected. This is the image of the selected site. So these are these parameters visible, sir? Hello, am I audible? Am I audible?
Yes, audible.
Okay, sir. So these are the parameters of the river. For the month of January, we studied different parameters, physiochemical parameters of Balol Tavi, like pH, conductivity, BOD, turbidity, TDS, COD, total alkalinity, hardness, calcium, magnesium, and chloride contents. So these are the parameters for the month of January. It means these are the parameters for the winters. So parameters, as we can say, the conductivity is very high, BOD is very screwed, pH is high, TDS is low as compared to the other seasons, as we see. So these are the parameters for the Balol Tavi for the month of April. We can say these are the parameters of the river in the pre-monsoon timing. So here the pH is quite high, like 8.1. Conductivity is also very high, BOD, and all these parameters are for the month of April, pre-monsoon season, we can say. So moving from this to, we get the parameters for the month of July. So this is, we can say, for the these are the parameters for the monsoon timing in the Jammu. So these are the parameters in the July. Coming to the next slide, let me share with you the parameters for the month of October. So we can say post-monsoon parameters of the water body. These are the post-monsoon parameters of the water body. So when we analyze these different parameters and we study them for the seasonal variation of this water body throughout the year, we found that Jammu Tawi recorded the highest pH in the month of December. That is 8.2. The highest conductivity was recorded in the month of January, and that was 640. And the lowest conductivity was recorded in the month of October, 380. Balol Tavi has the highest BOD of 6.5 in the month of April, and lowest was recorded in January, and it was only 2.2. In July, it recorded highest turbidity, that is 67, and the lowest was recorded in December, 44. Highest TDS was recorded in January, and the lowest was recorded in October. The highest TDS, that is total dissolved solutes, was very high in the month of January, and it was 367. And the lowest was also the lowest TDS was also on a higher side, and it was 224. And the highest COD, that is chemical oxygen demand, was recorded in April, and the lowest was recorded in January. The highest was about 36. So highest alkalinity was recorded in the month of July, and the lowest in October. Hardness was highest recorded in July, 380. The parameter was lowest in the month of October. Magnesium content was also very high in January and quite low in October. Chloride was quite high in October, chloride content, and quite low in, but it was not that low, but it was, as compared, it was found lowest in the month of October in Balol Tavi. And after studying these seasonal variations in the water body, it was found that all the parameters in the Balol Tavi were within the permissible limits of BIS and WHO, but they were not within the limits of, they were not within the acceptable limits. Some parameters were very, very screwed, like TDS was very high. If we compare it with the acceptable limit of BIS, but they were well within the permissible limit of the BIS. Thank you. That is all from my side.
Okay, so Sukhvinder Kaur, what are your findings? After that, you have to doing this research.
Ma'am, actually, sorry, ma'am, let me, actually, ma'am, I am trying to study the effect of wastewater and polluted water on growing Brassica gentiana. So we are studying the seasonal differences and the effect of these differences on Brassica, which I am still doing.
So what you have to
So
do?
as I am doing this, yes, ma'am.
What differences you find out?
Ma'am, it is still, I am still studying it. We are studying the effect of different wastewaters and the effect of the different parameters of wastewater on growing Brassica gentiana. I'm still doing it. I have not completed that experiment. It is in the process. For next time when we meet, I will definitely share with you what are my findings regarding the impact of this
Oh,
wastewater,
wait
different
a
seasonal variations on growing Brassica in it.
It's a national conference. We cannot wait for the next time, please. So what you conclude after that, when you have to doing this, you have to take the research paper. The research paper, what they says, you have to see. So each and every research is depend on the findings or the research gap. You have to understand the research gap.
Yes, yes, ma'am. I know there is a research gap, and I am working.
You have to tell me the research gap.
Okay, okay. Ma'am, I am working on it. I am working on it. These are just the study of the analysis of the different parameters and the seasonal variations in it. But still, I have not got the result for what effect it will have on Brassica.
Okay, no effect.
Yes. Okay.
Vivek Kumar.
Vivek Kumar.
Vivek Kumar, are you there? Vivek Kumar. Baljinder Kaur. Sukhvinder Kaur, you have to leave, please.
Okay, ma'am.
Rhythm. Rhythm, are you there? Then Dr. Suresh Kumar from Government PG College, Unnao. Nandlal.
Nandlal. Nandlal.
Nandlal. Nandlal. Are you there?
Nandlal.
Yes, ma'am. Yes, sir.
You have to share your screen and start
Share
the
your screen.
presentation.
Okay, sir. Okay. Hello, sir.
हाँ जी, हाँ जी.
Is my screen visible?
No, अभी तो नहीं कुछ है.
Hello, sir.
Is it visible, sir?
No, नंदलाल, अभी भी स्क्रीन आपकी शेयर नहीं हुई.
Sir, ये तो offline हो जाएगा.
नंदलाल अपनी स्क्रीन शेयर करो प्लीज।
एक मिनट सर, एक मिनट।
अरे सुरेश कुमार बैठे हुए क्वेश्चन
आशीष जी, इसमें स्क्रीन करना शेयर कैसे करेंगे? ये ना मीट।
Nalla, you have to wait for some time. We have to start with the next one. Savita Devi, Suresh Kumar, Deepika, are you there? Ritu, Divya, Suresh Kumar, Vivek Kumar, Parvinder Kaur, Janinder Pitambardar. Hello, Janinder Pitambardar, Ritika, Sheetal, Suresh Kumar, Vishal Mahi, Deepti Gupta. Okay. ये कौन है? शीशपाल का तो कल हो गया था ना?
आगे हुआ है, नहीं हुआ है मेरे शीशपाल नहीं हुआ, शीशपाल का हो गया लेकिन।
हेलो, गुड मॉर्निंग सर।
गुड मॉर्निंग।
गुड मॉर्निंग मैम। मेरी प्रेजेंटेशन हुई नहीं है कल, आज थी मॉर्निंग में, उस टाइम मैं क्लास में था अभी।
प्रेजेंटेशन कल था आपका?
हाँ कल था मैम।
नंबर क्या था आपका?
पोस्ट प्रेजेंटेशन।
सीरियल नंबर का
पोस्टर प्रेजेंटेशन करना है आपने?
पोस्ट प्रेजेंटेशन जी मैम।
ओके, पोस्टर प्रेजेंटेशन। हाँ, आप स्टार्ट कीजिए, आपके पास 5 मिनट्स है, यू हैव टू स्टार्ट नाउ।
ओके मैम।
शेयर योर स्क्रीन प्लीज।
आई एम जस्ट शेयरिंग। मितू आइए।
शीशपाल 3 नंबर
ला रही हूँ।
बड़ी
वीडियो नहीं
वीडियो भी कर ले
ना ना रेंज रेंज बस बस यार ये आ जाना है ये Am I audible, sir?
Yes, yes, you are audible.
Sir, my topic of presentation is impact of pH on quality of solution grown L-leucine hydrobromide single crystal, which is a promising analog material. In this paper, we made attempt to grow good quality L-leucine hydrobromide single crystal in normal condition as well as in optimized condition to get the better quality crystal. First of all, why to select this crystal? First of all, we can say that in a survey of world market, the maximum budget is allotted to the growth of the crystals, out of which we can say that though हमारे पास 10% of the budget of the single crystal is given to the non-linear optic crystal. And what is the here? So first of all, we synthesize the L-leucine hydrobromide single crystal, and we take L-leucine in a beaker dissolved in distilled water, and then we add it the hydrobromic acid, the aqueous solution of hydrobromic acid, and after that we use the slow evaporation solution technique to get the salt synthesized. And after the synthesized salt, the salt is dissolved as solubility of the L-leucine hydrobromide and kept it for a constant evaporation. In first attempt, we will try to grow the crystal at ordinary condition, but later on we try to grow the crystal as at different value of pH, because the quality of the grown crystal at ordinary condition is not good. That is as shown in this figure. It is only like a transparent film type hai, and as we change the value of pH to different values, and we have taken the 3 photograph of the crystal, as the pH value decreases, the quality of the crystal get increased, means the thickness of the crystal increases as well as the mechanical strength will increase. So the properties and the growth of crystal can be explained in terms of the metastable this diagram. L-leucine hydrobromide is an organic material, and it's synthesized in the solution by the slow evaporation solution technique growth, and the growth mechanism can be understood by these terms. In this curve, the area between these two lines represents the metastable zone width, and this is the lower line represents the solubility curve, and the upper line represents the supersaturated curve. And in between there's the metastable zone width hai, the growth process lies in between the metastable zone width. So जो synthesized salt है, that was analyzed by single crystal XRD, and the result of single crystal XRD are almost exactly matching with the report with the observed value of this crystal. The XRD recorded is shown here, and after that the WH Williamson Hall analysis was done and plotted in this graph. So here it confirms that the synthesized salt is of L-leucine hydrobromide crystal. The main features on the crystal structural parameter, the impact have given that if we have taken out the high resolution XRD analysis of the ground crystal, as the crystal which is of good condition is at 1.5 and the best is at 0.88 pH value. These are the high resolution XRD curve recorded between the glancing angle and the diffracted intensity, and here at the pH value of 1.5, the ground crystal shows that the full width at half maximum is around 75.29 arc per arc second. And when we can say when we analysis this with HR XRD with the crystal grown at pH 0.88, we have the full width at half maximum rapidly decreases and it reaches to 17.59. It means with the decrease in the full width at half maximum shows that there is a rapid increase in the crystal perfection. And further, if we see that the crystal grown at the aqueous solution of L-leucine hydrobromide have the pH value of around 4, and at around 4, the grown crystal is like only a film, and at 1.5, it's have some thickness, but the mechanical strength is so low. And further, we can say that the crystal grown at 0.88 and under optimized condition is very the high crystallinity, and that is full width at a uniform full width, a single peak with uniform intensity on both the sides with a FWHM 17.59 arc. And further, here we can also assume that the change in value of pH absolutely affect the growth quality of the crystal, and that the main parameters are that the value of pH also control the nucleation of the atoms or molecules in the solution. And further, we can say that the value of pH will also increase the induction time period of the growth of the crystal. And here, this is the hardness and etching study. As the hardness was also studied, and these are the some some micro picture from the Wicker micro hardness microscope, and these are used for each. After etching, before etching, the crystal surface lies like this. After 5 seconds, this looks like almost plain, and after then these hillocks were also observed at different time of etching. Etching is just the reversible process of the crystal growth. As the crystal growth, nucleation starts, and the atoms are getting close to each other to form the crystal, and etching is its reverse process. This crystal is very useful. This crystal is a nonlinear optical, and it has a very large value of second harmonic generation efficiency that is equivalent to approximately 4 times than that of the KDP crystal. And are much more, if we are able to grow this crystal with much more mechanical strength and also the good optical transmission, we can say that this crystal can be a potential candidate for like optical modulators and other electro-optic devices. Thank you.
Rakesh sir? Rajesh sir? Dr. Rajesh? Sir?
Yeah, जी मैम.
Presentation, so please you can.
Okay. Hello.
Yes, sir.
Shishpal ji, what is the significance of your study towards the societal benefit? Societal.
Sir, actually, we can use this impact of pH in order to get good quality of the crystal. And if these crystal which have the large value of second harmonic generation efficiency, this can enhance its use for photonic applications.
Okay, so means what is the benefit to the layman? एक common public को क्या फायदा हो सकता है? आप क्या मानते हैं?
हाँ, बिल्कुल सर. Common the benefit to the layman is that कि if जो coming era है, that is of the जो electronics to moving from electronics to photonic stage, and these crystals, this can be used in like aerospace application or like military applications or any other computer device, computer with photonic technology device. Its use can be expanded to this.
चलिए ठीक है, fine. ठीक है.
Thank you, sir.
Wonderful presentation. We can switch to next.
Thank you, sir. Thank you.
Here, any for the poster presentation? Any candidate for the poster presentation?
कोई पोस्टर प्रेजेंटेशन के लिए और स्टूडेंट है?
Yes, what's your name?
मैम जतिन कुमार।
जतिन, आपकी कल प्रेजेंटेशन थी या आज ही है?
सर कल प्रेजेंटेशन थी तो कुछ इशू की वजह से मैं दे नहीं पाया, तो अभी ऑनलाइन प्रेजेंटेशन देना चाहता हूँ मैं।
ओके, आपका ऑफलाइन में नाम था पहले?
यस सर।
Okay, you have to present.
ओके मैम।
शिशपाल जी अपनी प्रेजेंटेशन बंद कीजिए प्लीज।
Is this visible?
जतिन, इसको ये मोबाइल पर ही चल रहा है ना, तो इसको लैंडस्केप में करो।
ओके
और थोड़ा प्रेजेंटेशन को बड़ा कर दो उसके लिए। अब इसको बड़ा करो, थोड़ा और करो।
Sir, can I start?
Yes.
Yeah. The topic of my presentation is molecular marker and their application in improvement and production of root crops. Firstly, the introduction. Molecular marker are genes or particular segment of DNA that are representative of difference at DNA level. In genetics, a molecular marker is a fragment of DNA that is associated with a certain location within the genome. There are some properties of molecular marker. It is very simple process, inexpensive, with codominant inheritance, provide enough isolation of genetic variation, easy to assess, require little amount of DNA sample, and having relationship with adverse phenotype, and easy exchange of data among laboratories. There is a method of genetic diversity. Firstly, collection of plant material for the DNA extraction and storage. The second step is DNA extraction process. Third step is the DNA quantification by gel electrophoresis. The last one is PCR. Here is the photograph. Firstly collect the plant sample, then load the DNA sample. उसके बाद हम स्क्रीनिंग करते हैं, उसके बाद हम पीसीआर लगा के उसकी आइडेंटिफिकेशन करवाते हैं. नेक्स्ट वन इज application of molecular marker in fruit crops. The major objective of breeding program is improvement in development of new commercial and high yielding cultivars. Marker locate rates combination may possible be used as selection criteria for diverse parent selection and selection in segment segregating population during commercialization. Here is the application of identification of QTL. There is the some fruit and their traits. और उसके ऊपर ये वाले हमने मार्कर लगाए हैं और उसके बाद हमने उसकी आइडेंटिफिकेशन चेक की है कि किस हिसाब से उसकी आइडेंटिफिकेशन की गई है इस मार्कर के थ्रू. उसके बाद नेक्स्ट एप्लीकेशन है हमारे पास रेटियल आइडेंटिफिकेशन. इसमें भी हमने चेक किया है कि ये वाली क्रॉप्स ली हुई है हमने और उसके ऊपर ये वाले मार्कर्स हमने यूज किए हैं और उसके बाद हमने उसमें वैरायटियल आइडेंटिफिकेशन का चेक किया है कि ये वाली वैरायटी किस वाली वैरायटी से डिफरेंट है. इजीली हम चेक कर पाते हैं मॉलिक्यूलर मार्कर के थ्रू. उसके बाद नेक्स्ट एप्लीकेशन है हमारे पास मार्कर असिस्टेंट सिलेक्शन. इसमें इसमें डायग्राम के थ्रू बताया गया है कि ट्रेडिशनल प्लांट ब्रीडिंग में हम अगर कोई डोनर पेरेंट और कमर्शियल प्लांट वैरायटी लेते थे तो उसको दोनों का क्रॉस करवाते थे, तो जो डोनर पेरेंट जो हमें डिजायर्ड जीन चाहिए होता था वो उसके साथ अनदर जीन भी जो है वो नेक्स्ट प्लांट वैरायटी में ट्रांसफर हो जाते थे सिलेक्शन के थ्रू। जो हमें डिजायर्ड जीन चाहिए वही वाला हम सिलेक्ट कर वही वाला हमें जो न्यू इम्प्रूव्ड वैरायटी बनेगी हमारी, उसमें वही वाला सिंगल जीन प्रेजेंट होगा। उसके बाद ये कुछ अचीवमेंट है प्लांट ब्रीडिंग ऑफ फ्रूट क्रॉप्स थ्रू मोलिकुलर अप्रोच। इसमें सिट्रस, मैंगो, बनाना, एप्पल और ग्रेप्स ये वाले मार्कर लगाए हैं और इन पे ये वाला वर्क हुआ है। उसके बाद रेफरेंस है और इसके क्लियर है कि जेनेटिक डाइवर्सिटी एनालिसिस प्रोवाइड अ powerful tool that enhance a comprehensive understanding of genetic variation and improve conservation strategies. The ability of PCR-based molecular marker and next generation sequence allow for the detailed analysis and evaluation of genetic diversity in plant and to detect genes that influence certain desirable and important traits. It's all about you. Thank you, everyone.
So, Jatin, what do you think whether the molecular marker technique is much beneficial in comparison to traditional ones, or it is just deterioration of the crop system or agricultural system?
No, sir. It's molecular marker technique is very beneficial for identification of QTL भी उसके प्लांट्स में जेनेटिक्स में क्या डिफरेंस है, उसकी मोर्फोलॉजी में क्या डिफरेंस है, वेराइटियल आइडेंटिफिकेशन और असेसमेंट ऑफ जेनेटिक डाइवर्सिटी काफी इजीली वे में हमें डिटेक्ट करके बता सकते हैं।
वो जानकर हम क्या करेंगे?
वो जान के सर जैसे कि काफी टाइम लग जाता है किसी प्लांट को ग्रो होने में। कुछ प्लांट्स फ्रूट प्लांट्स है उसको जब तक वो बड़ा नहीं होता, जब तक वो फ्रूट हमें प्रोवाइड नहीं कर पाता, तब तक हमें उसकी वैरायटी का कई बार पता नहीं चल सकता। तो हम मॉलिक्यूलर उसके सीड से ही पता लगा सकते हैं कि बड़े होके इसका फ्रूट कैसा निकलेगा।
अच्छा, चलिए ठीक है। यू आर पीजी और पीएचडी स्टूडेंट?
सर
एमएससी स्टूडेंट। चलो, वंडरफुल। Presenting at MSc level is much appreciable. Thank you.
Thank you, sir. Thank you, sir.
Next one.
Now, in this session, anyone from poster presentation?
कोई और स्टूडेंट है जो इसकी पोस्टर प्रेजेंटेशन में ऑनलाइन हो?
No. Okay, now again I start with the oral presentation. Nitika, are you there? Nitika, Sheetal, Priyanka, Dipti, Vishal, Vivek, Deepika, Ritu, Divya, Suresh, Shivani, Nandlal, Ritu, Divya, Suresh, Vivek Kumar, Parvinder Kaur, Jininder, Pitambardar.
Yes, ma'am. Yes,
ma'am. Okay. Pitambardar, you have to start your presentation and your times are 5 minutes.
Pitambar Singh, हाँ,
yes, sir.
Share your screen, please.
Yes, sir. 1 second, sir. Sorry. One second, ma'am. 1 second. Is it visual, ma'am? Hello? I'm audible?
Yeah, you are audible, but your screen is not.
Oh, sorry. 1 second.
Share your screen, please.
Okay, sir. Okay. Is it visible, sir? Hello?
हाँ इसको फुल स्क्रीन कर दो आप इसको.
Okay, sir. Okay. It's visual now, sir. Hello. Afternoon all.